Hello, and welcome to the Disrupting PFAS podcast. I'm your host, Jeff Hale. For this season of the podcast, our theme switches to natural materials and processes for the detection, destruction, and sequestration of PFAS. Today, I'll be talking with Erlend Sørmo about stabilization of PFAS-contaminated soil with activated biochar. Erlend is affiliated with the Department of Environmental Chemistry at the Norwegian Geotechnical Institute and also with the Norwegian University of Life Sciences. I chose this topic because it makes use of natural material, biochar, that shows promise for the sequestration of PFAS. So let's learn more about the topic from one of its innovators. Welcome to the podcast, Erlend.
Thank you, Jeff.
So could you please tell us about the Department of Environmental Chemistry at the Norwegian Geotechnical Institute?
Oh, yeah, of course. We're a rather small group of about 15 people. We're mainly environmental chemists, some marine and geochemists as well. We used to be a part of a much bigger and more general environmental consulting department. We had a bit of a restructuring process a couple of years ago. So now we're kind of focused the chemists in one area. We do a lot of different things, both consulting and applied science. And we tend to go where the next challenge is. So this is why during the last 5 years or so, we've gotten quite heavily into microplastics and PMT-related issues such as PFAS. We also collaborate a lot with other groups at the Institute. So we often get involved in projects where geotechnicians or geologists maybe are, where the main issues are related to geotechnics and geology rather than chemistry. So yeah, we have a varied selection of problems to work on here.
Yeah, I noticed that from reading your publications, it looks like you have a broad experience with a range of geotechnical and environmental issues. I think I'm like you. I tend to gravitate to the hot topic or where the action is. Was there anything specific that drew you to PFAS other than it's a significant environmental concern these days and it's getting a lot of attention? Was there anything specific that drew you to that topic?
Well, yeah, I had this approach when I started here about 8 years ago that I would try to get involved with as many things as possible, try to learn as much as possible, hence the broad kind of range of topics I have in my publication list. But there were two things some years back that kind of coincided. The one thing I was working on a consulting project where I was doing a risk assessment for hazardous waste sites in Norway. And we discovered quite a lot of PFAS in the landfill leachates. And the customer didn't really know why or where it came from. They had no clue. So this led to a lot of interesting detective work. And at the same time, I got this, I stepped in as a temporary technical lead for contaminated sites, because my colleague Sarah Hale had a maternity leave. So in this role, I was in charge of leading these work groups on PFAS. And we had quite a lot of PFAS-related projects, and Sarah has been one of our pioneers when it comes to PFAS. And so she had set up this work group where we could discuss and share experiences related to PFAS projects because they offered a lot of challenges. A lot of things were quite different to other contaminants we've worked with. So through leading these work groups and trying to figure out this problem for my landfill client, I kind of started realizing how the scale of this problem and how sorely good solutions are needed. So this is I'm kind of drawn to challenge. So yeah, I guess this is why or how it started.
Okay, well, thanks for sharing your pathway to PFAS with us. I know we're dealing with the number of instances where PFAS are related to landfills and obviously PFAS are associated with a variety of industries. I think sometimes different industries or drivers might differ from country to country. So thank you for sharing your perspective from Norway. So I was reading NGI's proposal for new normative values for PFAS and PFOA in soil. One thing that I wasn't particularly clear on from reading the document was whether those values were protection of groundwater values due to leaching of PFAS from soil, or if they were just general soil criteria for PFAS and PFOA. Could you explain that further, Erlend?
Yeah, of course. I wasn't involved in this particular work myself. I've done some work related to deriving such values, and I also spoke to my good colleague Hans-Peter Arp, who was responsible for this, and he said that they had assessed 4 endpoints when they derived these values, and they were human health, soil ecotoxicity, aquatic ecotoxicity, and secondary poisoning. So in this human health endpoint, the leaching to groundwater is considered. Because the way we usually do it is that when you consider an endpoint and there are multiple exposure routes, or like for the human health issue, then you choose or try to identify the most sensitive one. And for human health, the exposure to PFAS and PFOA through drinking water wells was the most sensitive exposure route. And this was actually the one, the controlling value, was the endpoint that was chosen to control the normative value. So it does consider the PFAS leaches to groundwater, and then a human being drinking this groundwater. Actually, the aquatic ecotoxicity gave even more sensitive value, but it wasn't really applied here because it would be more relevant for special cases where soils are close to lakes or coasts and where there's little or no dilution occurring. So that would be like very specific cases. So groundwater is actually the key thing here. And this is like a general approach when when there are many different things you have to evaluate. You try to find the most critical, like the most sensitive, and then you use this as your guideline. Of course, the drinking water level, or the amount of PFAS before in drinking water, is then related to weekly tolerable intake. So that's how it all fits together.
Okay. Well, thanks for that explanation. I think your work on the stabilization of PFAS in soils can definitely play into that leaching to groundwater criterion and help, at least help to eliminate that pathway. Mm-hmm. So when we started talking, you told us about the Norwegian Geotechnical Institute, but I understand you're also affiliated with the Norwegian University of Life Sciences. Could you tell us a little bit about your work and role at the Norwegian University of Life Sciences?
Yes, of course. Well, the thing is that I'm currently working on a PhD, and I still have my job at the Norwegian Geotechnical Institute, where we have a big research project that is funding my PhD. But I'm affiliated with the university because they are, of course, the official institution that will award the degree when or if it's completed. So this work I'm doing now related to producing biochar sorbents and stabilizing PFAS, amongst other things, is a part of my PhD project.
Okay, well, thanks for that, and good luck in the pursuit of your PhD.
Thank you.
So why don't we switch and talk more specifically about some of your recent research. So today we'll focus on stabilization of PFAS-contaminated soil with activated biochar. So Erlend and his team have recently published that topic in the Science of the Total Environment, so congratulations to you and your team.
Thank you.
So I guess a good place to start, Erlend, is biochar itself. What is it and how is it produced?
Well, biochar is a porous carbon-rich material that you get when you take some kind of organic matter and heat it to a fairly high temperature in an oxygen-free environment. This process is called pyrolysis.
So what type of materials or feedstock, if you want to call it that, are used to create biochar?
Well, you can make it from anything really, as long as it has fairly high content of organic carbon. So, but typically biochar has been produced from agricultural residues. So, for example, rice husks or coconut shells or corn stovers or like any kind of surplus organic material that there's little or no other use for. But you could also make it from other types of materials like just wood or wood waste, and more recently people have also been looking into using sewage sludge, food waste, or other such waste materials. But there's an important discussion in the biochar world about this term and how it should be applied. Because some people say it should relate only to sustainable production chains. So you take some kind of waste material, preferably clean residues like the agricultural residues, and then you pyrolyze them. So you shouldn't grow a tree solely to cut it down and make biochar from it, because then you kind of ruin the whole sustainability perspective here. And also, a lot of people are saying that if you pyrolyze plastics, for example, or material or a waste fraction where there's a lot of plastics, you shouldn't be able to call it biochar because plastics is made from a fossil carbon source. Well, I myself, I'm not a purist. I think biochar should be any pyrolyzed organic substance, but I do think it's very important to consider the production chain and the bigger picture. So, like, consider emissions, and you consider whether you're cycling contaminants in the process or releasing them. And and you should really consider that the other potential uses for the feedstock. Like if there are other higher value chains that make more sense, you shouldn't be producing biochar.
Well, appreciate that clarification. I've been looking into biochar myself quite a bit. It is a fairly broad term. One thing that piques my interest is the opportunity to upcycle a waste material and then further use it as an environmental treatment medium, especially to remove PFAS. So I think the sustainability and upcycling potential for that type of material is very promising. But I do appreciate your clarification that it needs to be done sensibly, and not everything that's pyrolyzed would constitute biochar. Your definition sounds, or I guess the material itself, the biochar material, sounds very similar to granular activated carbon, which is commonly used to treat PFAS and other things. How is it similar? How's it different?
Well, as you say, it is quite similar. Biochar has been used a lot for agricultural soil improvement, but lately, or I guess during the last 10 years or so, or a bit more, people have started realizing this, what seems like the rather obvious connection between AC and biochar. Like you have a carbon-rich porous material that could maybe trap contaminants. And they are similar. The thing is that AC is usually produced from a fossil source. So you take lignite or coal, and then you activate it through some kind of activation process. And this usually gives a pretty uniform product. High internal surface area, high porosity, not a lot of exciting surface chemistry going on. You remove a lot of the functional groups in the activation process. It's like very condensed aromatic carbon. The properties of biochar vary quite a lot depending on the feedstock you use and the pyrolysis conditions. So, but if we should try to find the biochar type that would be most similar to AC, it would be a biochar produced from some kind of solid wood residue at a high temperature. And ACs are generally considered better sorbents due to these the high internal surface area and the porosity. And you can get this in biochars that are produced at high temperature, for example, or maybe activated like we did. But they tend to be to have a little lower porosity, a little lower surface area, not just the same structures. The thing is, though, that biochars have a very variable surface chemistry. So this could actually be a benefit in some cases if you want to, for example, trap heavy metals. It doesn't really work too well with AC, well, not for all heavy metals. But if you have a biochar with more functional groups on the surface, these could maybe aid in trapping heavy metals. So biochars are much more diverse than AC. So it's similar, but it's not perfectly similar.
So you touched on it a bit, but could you talk about the activation process? What is done to activate biochar?
Well, the activation process is an oxidation process. So you need some kind of oxidation agent. And this oxidation agent attacks the carbon, some of the carbon, and it removes the surface functional groups and it oxidizes and tears down thin layers of carbon. So maybe pore walls are removed, and this expands the pore structures internally in the coal in the terms of activating to make an activated carbon, the traditional one, and then biochar just expands the existing pores or create new ones. You can use chemicals to do this, or you can use physical activation at high temperature. So it's funny that we call it a physical activation because it's actually it's also an oxidation process. But it's if you use a chemical, you have to use strong acids and bases, and there's issue. You produce maybe waste materials like acid or base waste. Whereas if you do a physical activation, you add, for example, steam or water. A lot of people consider physical activation the best way to go. Physical activation can be done either in one or two-step operation. So in the one-step operation, which is the one we used in our paper, we add steam or CO2 directly into the pyrolysis chamber. So maybe this would give you a little pause and you're thinking like CO2 or water doesn't really sound like a very powerful oxidant. We could of course add oxygen, but then this is too powerful oxidant. You will eat away all the carbon. Of course, if you add too much O2, you get combustion instead of pyrolysis. So you add steam or CO2, but you have a very high temperature in your pyrolysis reactor. So typically more than 800, 850 degrees. And this high temperature kind of takes you above the activation energy needed. So the water or the CO2 can act as an oxidant. So this is how it's done generally. A two-step process. You take biochar already produced, and then you put it into a reactor where you won't do the same. But if you're doing a one-step process, you have to also pyrolyze the biochar at a high temperature, because then you're injecting the oxidant in the pyrolysis process. And the two-step process, you can take biochar already produced, maybe at a lower temperature, and then activate it later. There's not a lot of research about the pros and cons of choosing the one method over the other, but maybe someone will tell us more about this in the future.
Okay. An interesting aspect of your paper that I thought was the optimal balance between the amount of biochar amendment, the degree of activation, and the production yield of the biochar. So I think those three variables, it's a balancing act to get the right proportion. Can you talk about those variables and how they relate to each other? So the amount of biochar you would have to use in an amendment compared to its degree of activation and how that relates to the production yield of the biochar?
Yeah, that's a very good point. This is one of the things I was thinking quite a lot about when I wrote this paper. The thing is that the amount of biochar amendment needed depends on how high the affinity the biochar has for your target contaminant. So if you have a weak biochar sorbent, you need a lot of it. And of course, this comes at a cost. You have to produce all this biochar and the energy that goes in the process and the feedstock needed, so on and so forth. But if you make biochar at a low temperature or non-activated, it gives you a higher production yield. So for the case of PFAS, producing a weaker biochar will give you a higher yield, so you get more of it. And you also retain more of the carbon in this process. But if you activate, you get a much stronger sorbent because you expand the pore systems. But and then you need less of this sorbent because it's stronger. This process, of course, requires more energy and it reduces the production yield quite a lot. So for the biochar we produced for our paper, I think the yield was almost halved by going from non-activated to fully activated. So you lose a lot of material in the activation process because, as I said, it's an oxidation process. So it's very important to balance your production between making a lot of a weak sorbent or less of a stronger sorbent. So I think it's quite important to consider your amendment target. So it could be that your remediation operation does not require a very strong sorbent, and it's okay to produce a weaker one. But if you have a very tough case, then maybe you need to go fully activated high doses. So yeah, it's a bit of a trade-off to be considered there, and I think it's probably best solved using a life cycle approach. Get someone to do the life cycle analysis.
Yeah, I think it's important for people to be aware of those variables and how they affect each other. But you also added the fact that it kind of depends on your objective or the type of site you're applying the material to. So if you had a very small volume of impacted soil, perhaps at a high concentration, that might require more activated biochar at a lower volume. But if you had a larger site, maybe there would be a trade-off in more material at less sorptive capacity. So that's something that I think was a good add on your part. Appreciate that. So let's talk about PFAS-contaminated soil. So your paper and your study focus on amending PFAS-contaminated soil with biochar. What type of soils were used in the experiments?
Well, we got our samples from an old firefighting training facility at a decommissioned airport in southern Norway. So we used both the topsoil, which was very organic rich, 34% TOC, if I remember correctly. And we also sampled the lower lying mineral soil, which had very little organic matter, so about 1.6%. And this was a typical example of soils affected by legacy contamination of AFFF, firefighting foam. So they've been putting out fires there at this training facility and spreading all this foam into the soil. This is, I guess we have a lot of these sites around the world. But this was typical old AFFF with mostly PFAS. It was 83% PFAS in topsoil and 89% in the lower lying soil. Then you had a smaller fraction of the PFAS, which was 5 and 9% in the top and lower lying soils. And I think the PFAS concentrations were 1,000 and 3,400 micrograms per kilo in the top and lower lying soil. So this was quite high. It was, I think, about 1,000 times higher than this normative value we're talking about earlier.
Well, that's probably a good transition to the next question. So I'm looking mostly at PFOS and PFHXS in soil at concentrations 1,000 times the normative value. And so let's talk about the results. How effective was the biochar at sequestering the PFAS?
Well, it was actually surprisingly good. We're very happy to see the effect of our sorbents. And we look, as I said, our main focus was, of course, on PFAS and PFHXS, since they were the most abundant. But we also had a range of other PFAS, such as PFOA, I think it was 1% or 2%. And then there were other PFCAs and PFSAs and a couple of the fluorotelomers as well. So there was like, we have data on a lot of different PFAS, but of course the focus was on PFAS mostly. I think in some of the scenarios we tested, we had almost 100% retention of the PFAS, but it did vary quite a bit. And we identified several factors that affected absorption. For example, in the mineral soil, we got really good results either with a high dose of the non-activated sorbents or with a low dose of the activated ones. It was much more difficult to get good results for the high organic topsoil, however, because for this soil we needed a high dose fully activated sorbent to get close to full retention. And also it was much more difficult to retain some of the shorter chain PFAS compared to the longer chain ones. So for PFAS, we got really good retentions, but for PFBA, for example, it wasn't that good, given at fairly high doses. It wasn't perfect. Yeah, so it's complicated, and this kind of brings us back to targeting sorbent for a specific case. So here, we see that the soil organic matter matters a lot, and also the type of PFAS you have in your soil. So it's not an easy thing to solve.
Yeah, we see that with a lot of sorptive media. The challenge of sequestering or retaining those shorter chain PFAS. And maybe that's a good segue into my next question. What are the challenges, limitations, and further research that should be conducted in this area, in your view?
Well, as you mentioned, these short chain PFAS are difficult to retain, and we need to find out more about the mechanisms that control the interactions between these short chain PFAS and sorbents, in order to optimize the retention of these PFAS. And what has been found is that hydrophobic interactions between the fluorocarbon tail of the PFAS and the surface of your AC or biochar is the main sorption mechanism. But you also have some electrostatic interaction between the functional heads and charged sites on the surface of the AC or the biochar. The thing is that the functional head is usually negatively charged under most ambient pH levels, and the same goes for the biochar surface. So you have a repulsion. For the long chain PFAS, you have strong hydrophobic sorption, and the sorption is so strong that the head group interaction doesn't really matter. But for the short chain ones, the repulsion starts to or weakens the hydrophobic interactions to such a degree that they're difficult to capture. So we have to find some ways of maybe modifying our sorbents so we can better trap these short chain PFAS. And of course, we need to upscale from lab tests to actual in situ stabilization to see how this works in field conditions.
Yeah, absolutely. Are there other use cases for the sequestration of PFAS with biochar? So we're talking here about amendments in soil, but I'm thinking about, are there use cases where biochar could be used for water filtration or as a permeable absorptive barrier? Do you envision that at all?
Absolutely. I think in theory you can substitute biochar into any process where you use AC, as long as the biochar in question has like a high enough affinity for your target contaminants. But you might have to do some work on the physical properties of the biochar material, because, for example, when you have granulated activated carbon, it has a nice, well-graded size distribution that allows for a nice water flow through a filter, for example, and allows for a certain kinetic uptake. So in order to make a certain biochar work for a water filter, for example, you need to think about maybe producing granules or looking into how the size and distribution of the biochar grains affects kinetic and hydrogeological factors.
Okay, I mean, I like the idea of diverse uses for this material, and we need as many solutions for PFAS that we can find. And I think a unique aspect of this, or an exciting aspect of this, is being able to use natural materials, especially if they're waste materials, for the absorption and sequestration of PFAS, be it as soil amendments or for water filtration, like other materials have been used. So that wraps up this episode of the Disrupting PFAS podcast. Thank you to Erlend Sørmo for joining us today. And I'm your host, Jeff Hale, reminding you to never say forever.